Insecticide resistance is one of the major factor which is adversely affecting pest management all over the world. At the time of invention of DDT, humans thought that we have solved the problem of insects. But within a span of about 8 to 10 years, the insect fought back and they demonstrated that they can survive whatever insecticide humans can discover. So that's one thing which evolved. The evolutionary capacity of insects has attained over the years. They have the innate ability to resist whatever kind of uh, insecticides that we discover. So insects will develop resistance, whatever be the mode of control which we adopt. So there are instances, recordically there are instances where insects have evolved resistance to cultural control also. Don't think that insects are evolving resistance to insecticides alone. There are, if you think about other management practices also, insects are evolving resistance to other management practices also. So today we will be seeing about what is resistance, insecticide resistance, what are the factors favoring the development of resistance, how a resistance develops, what are the factors contributing it, then what are the mechanisms of resistance. And today we will be seeing only about a part of the mechanisms of resistance, because mechanisms of resistance is a huge thing. I won't be covering the entire thing in today's class. Tomorrow I will be taking the rest of mechanisms of resistance. In the subsequent class, we will see about detection of insecticide resistance and ways to manage insecticide resistance. So this will be the schedule which we follow for this topic, insecticide resistance. Uh, so coming to insecticide resistance as such, if you see the definition of insecticide resistance, we can see that uh, insecticide resistance, there are several definitions, but I prefer the definition by WHO. Is it visible for you? Yes, okay. So, the insecticide resistance, WHO has defined as development of an ability in a strain of insects to tolerate doses of toxicant which would prove lethal to majority of individuals in a normal population of a species. I will repeat it again. It is the development of an ability. That means the resistance is developing inside the insect. Not all the insects are not, don't think that all the insects are born resistant to a particular kind of insecticide. Because of the repeated exposure to a particular pesticides, insects are evolving ability to resist the toxicity of insecticide. So that is development of an ability in a strain of insects. That means if Helicopa armigera from one part of India, or say from if you say from Karnataka, has evolved resistant, even in Karnataka, say Dharwad or Bangalore or Mangalore, so whatever it may be, whatever be the location, may have evolved resistance. But Helicopa armigera from another part of the same state or same country may not have evolved resistance. So that's why it's a development of an ability in a strain of insects, not all the entire insect species, in a strain of insects to tolerate doses of toxicant. That means the ability it has evolved is to tolerate the doses of toxicant. That means it will survive the pesticide application. So this is what we are saying is this is in comparison to a susceptible normal population of a species. That means we have two populations, two Helicopa armigera, one from Kerala and one from Karnataka. So Abhishek, what is your native place? Tumkur. Okay. say we have helicopter armadura from Tumkur, and we have you collected some helicopter from Kerala. So in Kerala, helicopter is not a major pest, and we don't normally go for any chemical management of helicopter. And if we compare the susceptibility of helicopter from Tumkur, Abhishek has brought some helicopter population, and we are comparing it with the susceptibility of helicopter from Kerala. So if in Tumkur area, in the area from where Abhishek is coming, where the farmers are repeatedly using some kind of pesticide, the helicopter population which Abhishek brought from Tumkur will exhibit some level of resistance to the pesticide when compared to the population from Kerala. That is what it is told, that it will prove lethal to majority of individuals of the normal population or the susceptible population of a species. So resistance, I will repeat it again, it is the development of an ability in a strain of insects. That means not all individuals of a species has evolved resistance to tolerate doses of toxicant which will prove lethal to majority of individuals in a normal or susceptible population of a species. So this is the definition which I prefer and which I use to explain to students. Then again, we have several definitions of it, and whatever be the definitions, it is almost one and the same, except for Iraq definition. In Sarik in 1987 has given a definition that resistance marks a genetic change. So that genetic change is what defined in the first part of WHO definition itself, development of an ability. That means a genetic change. The resistance marks a genetic change in response to selection by toxicants. That means that is it is tolerating doses of toxicant that may impair control in the field. So that is one Savik, that two definitions are almost the same. Savik's definition is resistance marks a genetic change in response to selection by toxicants that may impair control in the field. Iraq definition is slightly different from the above two. A heritable change in the sensitivity of pest population. That part is same with the WHO definition and SAVIK definition. That is reflected in the repeated failure of a product to achieve expected level of control. That also is there in the earlier two definition. The next part differs from the earlier definitions when used according to the label recommendation for that pest species. So Iraq has given a slight change in the definition, because Iraq is a body comprising of pesticide companies. So according to the pesticide companies, if someone is using a pesticide which is not recommended in its label, that means which doesn't have any label claim, then it cannot be considered as a resistance development. So that means if you take what is label claim, I will explain it when the registration, when we take up registration and all. If you take any a leaflet which accompanies any pesticide packing, you can see a table wherein there will be the crops in which description of the crops in which the pesticides are recommended, against which insect pests, and at what dose the pesticide should be used. So there will be a leaflet for that. So that represents the label claim. So if a company has not given any label recommendation for a particular insecticide to be used on a particular crop against a particular insecticide, and if a farmer has used that pesticide in a crop against a pest which doesn't have any label recommendation, and if the pest has evolved resistance, then Iraq says that it can't be classified as insecticide resistance because they have not recommended the use of that particular pesticide against that insect pest. So that is only difference between the definition of insecticide resistance by Iraq and other two. Then the basic thing with respect to all this thing is resistance is a heritable character. The WHO definition says that it is a development of an ability. Savik says that it is a genetic change. And Iraq says that it is a heritable character. So all the definitions agree that it is a heritable character. Remember, research evidences slowly coming up which says that resistance development may not be a heritable factor also. We can't say that resistance development is a heritable factor always. We will see in the last part, we will see how there are examples wherein insects develop resistance without any change in the genetic makeup. And because of the change in the genetic makeup of the insect, the plant protection tool, that means mainly we are discussing about the insecticide. The insecticide will be failing to control or manage the insects. So resistance development will lead to the failure of a plant protection tool. Here we are discussing about insecticide, tactic or strategy. So resistance development will lead to the failure of a plant protection tool, tactic or strategy. So this is with respect to the definition of insecticide resistance. Do you have any questions with respect to it? Yes or no? No. Okay. So, and few more things with respect to resistance is one is pre-adaptation, that is heritable character. That one thing we will say that we can take the example of corona itself. So, in our lab, uh, our the person which got COVID positive in our lab visited one of the earliest staff working in our lab itself. So all the other we have about 4 to 5 staff in the lab. So three of our existing staff went to the residence of the earlier staff for a courtesy visit, and they one that family got COVID positive after few days. Among these three persons which went to the family, only one person got COVID positive. So this means that one person got COVID positive and other two have didn't contracted the disease. In the same manner, if you check it, if a person in the family got COVID positive, we are seeing instances that other members of the family may remain uninfected with the virus. What contributes to it? There can be several reasons. One is inherent immunity, and another thing is that by chance the other persons may not have much interaction with that COVID positive fellow. So one other reason may be that at least a few individuals of our species, human beings, may have the genes resistant to the coronavirus. So that will be there. Every population will have diversity of genes wherein one individual may be resistant to a particular kind of infection, another will be susceptible to a particular kind of infection. So that's typical with our student life also. During our student or college life, uh, we usually have chickenpox infection in our school or college. So if you check it, several members of your class or your classmates may be contracting chickenpox infection, but there will be a few individuals which may have had interaction with the infected chickenpox infected fellow, but still doesn't contract the disease. Why? One reason may be that person may be have a gene resistant to the which contribute resistance to chickenpox. In the same manner, if you take a particular group of insects, particular species of insect, all the insect individuals of the same species may not be uniform. It have genetic diversity. So a few individuals of the species may have genes which can provide resistance to insecticide. So this pre-adaptation theory, this is a theory with respect to insecticide resistance, which says that the genes controlling resistance is already present in a population, insect population, even before the introduction of insecticide. That is why DDT was introduced in 1937 and that 39 as an insecticide. by the year 1947, houseflies has evolved resistant to DDT. How come? How quickly? Within 9 or 8 years, it has evolved resistance to DDT because a certain individuals of houseflies had the gene even before the introduction of DDT. That's why it is called as pre-adaptation theory. And how it happens? When an insecticide is, I will use a picture to explain it. See, this is the usual picture which we use to depict how resistance develop in an insect. See the case. See here. This is the first generation wherein we are introducing the insecticide. We have nine insects: one, two, three, four, five, six, seven, eight, nine. Of which one is having a slightly different genetic makeup, and we are spraying an insecticide, and after the spray of pesticide we don't expect all the insects to die. If you have 100 insects, don't expect that 100 insects will die because of pesticide application. At least 10 to 20 will survive. So here three of them survived, and of this three, one is having again having this genetic makeup which is contributing resistant to the pesticide application. Coming to the next generation, third, fourth, or fifth generation, these individuals breed, and we will have more individuals with the same genetic makeup. See, one is having red color here. This population had only one individual having the resistant gene. In the subsequent generation, we are what we are doing is we are killing all the susceptible individuals, and the remaining individuals will all be having resistant genes. And these individuals with resistant gene will mate, and slowly the number of individuals having resistance genes will increase. So by the time a fifth or sixth or seventh generation, we will be having huge number of resistant individuals. This is how we say that resistance has evolved over time by the exposure of to a pesticide. And we usually use a term called as frequency of resistance. Here we can see that 1 in 9 individuals is having the resistant gene. But say the case here. Here we have totally 5 insects, and among the 5 insects, 4 are having resistant gene. That means 4 in 5 is having resistant gene. So here the frequency is 1 in 9. Here the frequency is 4 in 5. So that is what we call about frequency of resistant gene. Another typical example is the same thing. Say we are having 1, 2, 3, 4, 5, 6, 7 insects. We are going for a pesticide application, and two individuals are having resistant genes because of the pesticide applied. All the susceptible individuals died. Only the surviving individuals will be having the resistance gene, and these two will breed, and the next generation will all be having resistant genes. So it will develop resistance to pesticide. So this is how the pesticide resistance occurs. So an insecticide which act as a selecting agent for the selecting individuals with resistance gene. That means we are having 7 individuals over here, and by the application of the pesticide we are selectively killing all the susceptible individuals and allowing only those individuals with resistance gene to survive. So that means it is a selective agent, kills all the susceptible individuals, allowing only the resistant individuals to survive. So it selects only the resistant genotype. So that is with respect to insecticide. Then coming to a brief introduction or the history with respect to insecticide resistance. The first report of insecticide resistance was in way back, about more than a century back, by Melander in 1914, wherein he has reported resistance in Sanjo scale, Quadraspidiotus perniciosus against lime sulfur, an inorganic pesticide. And as I told you earlier, in 1947 itself, houseflies evolved resistance to DDT, and till now, that statistics may have changed, but this is from a book which was published in 2018 or 19, wherein they have given a statistic that in the whole world we have about 574 arthropod species. Don't think that it is having insects alone, because vast majority of mites have evolved resistance to insecticides. So because of that, we are including arthropods also. About 574 arthropod species has evolved resistance against 338 pesticides. So that means that a single species has evolved resistance to multiple pesticides. So that is where, and the major orders which has evolved resistance is, one is Diptera, which has evolved, which is contributing 27 percentage of the whole arthropod species which has evolved resistance. Can you say which is the insect which is contributing to the maximum insecticide resistance among dipterans? Any guess? Because here we know, if you I ask which is the order which has evolved resistance, mean you might say about hemiprins and lepidopterans, but the data says that order diptera is the group which has evolved maximum resistance. Which insect among order diptera is contributing to it? Any guess? Wild guess? Housefly? No. No. Yes, Shiga is correct, that is mosquito. Because mosquito is the one which has evolved maximum resistance. That's why we have the largest insecticide development in Diptera, followed by Lepidoptera, that understandable, then Hemiptera, Coleoptera, and then mites. That's why I told you about arthropod species. And the major families which has evolved resistance is, one is Tetranychidae, red spider mite, then Quilicidae, that is mosquitoes, Noctuidae, we have Spodoptera, Helicoverpa, etc. Then aphids. And the major species are Tetranychus urtica, diamond magmoth. Diamond magmoth is a pest which evolves resistance pretty quickly to almost all the insecticides. Then Mysus persica, again an aphid species. Bemisia tabasi, whitefly, quickly developing resistance. Then Helicoverpa armigera. Those the values in brackets. Tetranychus urtica means it has evolved resistance to 93 pesticides. You can imagine India hardly about 280 something pesticides are registered, and it has evolved resistance to 93 of it. So, what is the power of an insect to evolve resistance? Diamond magmoth 82 pesticide, Mysis persica 74. So, this is the capacity of the insect to evolve resistance. And if we see which are classes of insecticides to which insects have evolved resistance, the major group to which insects have evolved resistance is organophosphates and carbamates, acetylcholinesterase inhibitors. But I think this level must have gone down because the use of OP insecticides and carbamates are decreasing day by day. And the second position is with sodium channel modulators because of the presence of pyrethroids. Pyrethroids are widely used for the management of mosquitoes, and mosquitoes are evolving resistance pretty quickly to pyrethroids, and they contribute 32%. Then GABA-gated chloride and fipronil they are contributing to 14%. Carbamates, again acetylcholinesterase inhibitors, that percentage, it may not be 7%, it will be less than that. And neonicotinoids, it is 4%, but invariably it will be high now because day by day neonicotinoid resistance is reported. And coming to our country, the first report of resistance was by Dr. S. Pradhan. Have you heard about had the name of S. Pradhan? Yes, where? Posabin. Okay. Ah, pesticide umbrella and all. Yes, yes, yes. Okay. Srisha, are you able to follow, or do you have any issues in network? Srisha, are you there? Okay, let's see. We'll continue. So then he was the first person to detect resistance in insect called Singara beetle. You might not have heard about Singara beetle. It's a minor pest in one of the crop called Singara. That is English name is water chestnut or something. I forgot. And the insect's name is Gararosella birmanica. And in our country, we have reported resistance in 17 field pests and 8 storage pests. But I feel the report is very few in number because we don't normally work on certain insects. Say, take the case of rice. A large quantity of insecticides are used in rice, but we don't normally screen this rice pest for resistance because if we want to screen a pest for resistance, we have to rear those pests in large numbers. Artificial rearing of rice pests in artificial diets is extremely difficult. We don't have much artificial diets which survives, which will be able, which can be used to rear rice pests. So because of that, we don't normally conduct any bioassays with rice pests. So it is a very low figure. It will be extremely huge number compared to the other compared to the 17 and 8 which is reported already. And usually people say that I have sprayed a pesticide and the insect has not been managed. The pest is still remaining, and usually people say that the insect has evolved resistance. Before making a statement, a blanket statement like that, make sure that we have identified the pest correctly and the correct insecticide was used. Because nowadays we are using specific pesticide, not broad spectrum insecticide. If you are seen a symptom in any crop and that problem was because of a sucking pest, and you have recommended a diamide pest for managing it, invariably the sucking pest will survive because diamides will manage only lepidopteran pests. So you have incorrectly identified the pest and a wrong pesticide was used. Because of that, the pest will survive and that cannot be calculated as resistance. And the next is the correct dose and proper method of application was used. Correct dose means say if you are recommending at the rate of 2 ml per liter and the farmer has used 1 ml per liter and if the insect is not killed, we can't say that it is because of insecticide resistance because that's because of the faulty calculation of dose or faulty application of the pesticide. And another is proper method of application. We have seen certain instances wherein farmer has gone for mixing a granular pesticide with water and spraying. I had certain instances when earlier carbofuran and forate was available for pest management. We had certain reports wherein farmer mixed a granular pesticide in water and applied and complained that the pesticide has not worked. Granular pesticides are not meant for mixing with water. It won't give any results. So we should follow proper method of application. Then we should check for the pest population. And, and the next thing should be the pesticide should be applied at the right time. Right time means that when we are applying the pesticide, the pest should be present. And another thing is that the pest should be at the susceptible stage. That means you have seen Spodoptera larvae feeding on the crop, and after 7 days you are going for a pesticide application. You didn't get the labor to go for pesticide application, and after 7 days you are going for pesticide application. By that time the larvae might have pupated, and pupal stage is one of the most resistant stage to insecticide. And if you're spraying the pesticide and after 1 week you are again seeing the Spodoptera infestation, you can't say that it is because of insecticide resistance. It's because when you applied the insecticide, the pest was not at its less susceptible stage. And we should make sure that the pest is present in the field when you are applying the pesticide. Another thing is that normally what happens is that people usually complain that mainly with respect to white flies and all. Sir, I have applied the pesticide which you recommend for managing white flies. It was effective for about 7 to 10 days, and after 2 weeks, I am getting white fly infestation again, and that's due to resistance. No, it's not due to resistance. The population decreased, and whitefly will migrate from another area, from untreated area to this our cropped area. When the toxicity of the pesticide residues decreases, the insect will migrate from another area to this area. So there will be reinfestation invariably. So this reinfestation may not be considered as, should not be considered as pesticide resistance. So we should make sure that we have identified the pest correctly, we have the correct recommendation for that particular pest, ensure that the exact dose is used and the proper method of application is used. And when we apply the pesticide, we should ensure that the pest is present and the pest is at the susceptible stage and there is no reinfestation of the pest after application. So only after confirming all these things, we should say that yes, there is a chance of development of insecticide resistance. And what are all the factors contributing to the development of insecticide resistance? The development of insecticide resistance depends on frequency of resistance gene in the population. See, I will show this picture. See, here I told you that this frequency was 1 in 9. And if the frequency is 4 in 5, we can say that this population will evolve resistance pretty quickly. So the frequency of resistance gene in a population is one of the critical factor which favors the development of insecticide resistance. Higher the frequency of individual with resistance gene, greater will be the rate of development of insecticide resistance. And next factor is the nature of the gene present in the insect population. The single gene may be contributing to the insecticide, multiple genes may be contributing, and you might have studied about dominant gene, you might be studied about recessive gene. I'll ask you a question. If the resistance is contributed by a dominant gene and in another insect resistance is contributed by a recessive gene, which uh, factor will favor the development of resistance quickly, dominant gene or recessive gene? Dominant dominant Sri Lakshmi dominant. Abhishek? No. Abhishek, are you there? Maybe recessive. Why? Why, Abhishek? What prompted you to, you can unmute and tell what contributed, what prompted you to tell a recessive gene might be contributing? You can go back to genetics class. What is the ratio of dominant versus recessive individuals in the F1 generation? 3 is to 1. That means 3 individuals will have which gene? Dominant or recessive? Dominant. And only one individual will be having the recessive gene. So that is why we call the dominant gene, if the insecticide resistance is due to dominant gene, then rate of development will be pretty quick. Insecticide resistance, but we don't normally see a dominant gene responsible for insecticide resistance. We don't normally see. So that is with respect to the nature of the. And then next is intensity of selection pressure. Intensity of selection pressure, that is selection is pressure is by because of the presence of insecticides. If the farmer, if a farmer is going for application of pesticide 10 times in a crop, and another farmer is going for application of three pesticides 3 times in a season, which which farmer will have the resistance development quickly? One farmer is going for 10 sprays in a season, another is going for 3 sprays. Which farmer 10 times? That's the intensity of selection pressure. The selection pressure is more, then resistance development also will be higher. Then rate at which species breed. Another thing is, uh, one thing I told you is DBM has evolved pretty quickly. And does and another insect pest we can take example is say root grub, Leucophonus coleopora or Holotrichia consanguinea. If we compare the to DBM and root grub, which one is having the highest life cycle, longer life cycle? Root grub. Okay, so which one will develop resistance quickly, DBM or root grub? DBM. Why? Because if you spray a pesticide, the pest will the resistance individuals will survive, all the susceptible individuals will got killed, and the the resistant individuals will breed, and the next generation will be there in the field within 1 month, within 3 to 4 weeks, because DBM the life cycle is hardly 3 weeks. So within next month we will have another set of resistant individuals. So that is the way the resistance develop pretty fast in the case of individuals which is having extremely short life cycle. So normally we don't get resistance development in say rhinoceros beetle, red palm weevil, root grub, etc., because they have extremely long life cycle. The insects evolve resistance pretty quickly because of its short lifespan. So that is one thing. And the next one is population mobility. Say, I will ask another question again. Uh, we have say aphids infesting one crop. I will take example of say aphids infesting bitter gourd, and in bitter gourd another major pest is fruit flies. So we have taken two pests. Which one is fruit flies and aphids? Which one is more mobile, fruit fly or aphid? Fruit fly. Okay. Which one can develop resistance quickly, fruit fly or aphid? No. Abhishek and Rahul, Srilakshmi all told fruit fly. No, it won't be fruit fly. Because what happens is, I will take the example picture early. Okay. So take this example. So here we have 7 individuals. We have of which 5 are susceptible, 2 are resistant, and we are going for a pesticide spray, and all the 4 individuals got killed. Only the 2 individuals with resistant genes survived. Take the example aphid and fruit fly. First, we will take the case of aphids. Aphid is a sessile individual. So aphid will remain there. It will breed. It will breed among themselves, and all the next generation will be resistant individuals. Take the case of fruit fly. It is extremely mobile. When all the individuals get killed, fruit fly, because as it is a mobile pest, once there is a reduction in population of fruit fly population in one side, fruit fly from another area, wild population of fruit fly will migrate to this area. This wild population will invariably will be having more number of susceptible individuals, and these susceptible individuals will breed with this resistant individuals, and the next generation will be again a mix of susceptible and resistant individuals. So that's why those insects which have mobility, higher mobility, has less chance of development of resistance, because after the pesticide spray there will be an influx of or the inward movement of wild population carrying the susceptible gene, and it will dilute the resistant gene frequency in that population. Are you clear? Yes. Okay. So that is with respect to the mobility. Then persistence. So that is one thing which is clear. That means if the pesticide is extremely persistent, then the resistant development will be faster. That means if you have a pesticide which persists for about 45 days, and if you are having a pesticide which is persisting for 7 days, that means that the pesticide which is having a persistence for 45 days, it is as if we are continuously applying pesticide for 45 days together, because that selection pressure is remaining in the field for 45 days. But if the pesticide is having short persistence, the persistence will be remaining only for an extremely short period. So because of that, the persistent chances of development of resistance is low with less persistent insecticide. So these are the factors which contribute to the development of resistance. One is frequency of genes, then the nature of gene present, then the intensity of selection pressure, then what is the rate at which the species breed or the number of generations per year, then the mobility of the pest and the persistence of the pesticide. So these are the factors which contribute to pesticide resistance development. This is another, see here we have 9, of which the frequency is 2 in 9 after pesticide spray. Only 4 surveys, of which 2 are susceptible and 2 are resistant. And in the next subsequent generations, they breed and they will give more number of resistant individuals. That means the frequency will be high. Again, it will lead to more individuals with the resistant genes. And this is with respect to frequency. How many individuals are having resistance genes? I haven't taught you about the graphs and all. Then so I am not discussing about the shift in graph and all. I will discuss it when we conduct our practicals. And there are two terms in the case of resistance. One is cross resistance and multiple resistance. Cross resistance means that we are spraying one insecticide. Take the case malathion. A farmer repeatedly uses malathion for the management of, say, Spodoptera, and after 3 to 5 sprays, he is feeling that yes, this which insecticide I told, malathion or quinarphos? Malathion. Okay. So the farmer repeatedly sprayed malathion. For after 5 sprays, he feels that malathion is not giving any effective control. So he shift. He goes to the shop and takes another pesticide, and the shopkeeper gives him chlorpyrifos. Okay, and he goes for chlorpyrifos spray. But with chlorpyrifos spray, even with the first spray itself, it is not giving any desired result. That can be due to cross resistance, because malathion and chlorpyrifos both are having the same mode of action, and the pest will develop resistance to another pesticide in the same group with the same mode of action, even if the insect has not been exposed to the pesticide pesticide which is used second. Malathion was used. It is an OP pesticide acting on acetylcholinesterase. The farmer has not used chlorpyrifos any time before. Since malathion was not giving any satisfactory control, he shifted to chlorpyrifos, but the insect is not getting control with even chlorpyrifos. So that is even though the insect has not been exposed to chlorpyrifos, that is cross resistance. Even if the insect has not been exposed to other pesticides, the other pesticides in the same group, the insect has evolved resistance. Coming to multiple resistance, as we took the example of malathion, repeated spray of malathion, it was not giving any control of Spodoptera litura, and he went to the pesticide shop, and the dealer gave him flubendiamide. Okay, it is Spodoptera, you use flubendiamide, it will control your Spodoptera problem. And he is using flubendiamide for the first time, and even after the spray of flubendiamide, he is not getting any desired control of Spodoptera. That means the pest has evolved resistance to another pesticide which is having a different mode of action, even though the pest has not been exposed to that particular pesticide. That is multiple resistance. So cross resistance occurs within the individual pesticides having same mode of action. Multiple resistance occurs if the pest evolves resistance with two pesticides having different mode of action, even if the insect has not been exposed to the second group of pesticide. Are you clear? Okay. Coming to the mechanisms of resistance, we can classify the mechanism of resistance into four. The first one is physiological, second one is behavioral, third one is symbiont mediated resistance, and the fourth one is insecticide resistance in parthenogenetic insects. So we can basically classify the mechanisms of resistance into four: physiological, behavioral, symbiont mediated resistance, and resistance in parthenogenetic insects. Among these four, physiological mechanisms are the major contributing factor to insecticide resistance, and we will see the physiological mechanisms of resistance in detail. And these physiological mechanisms of resistance are further classified into four. The first one can be the resistance can be due to reduced cuticular or gut penetration. It can be due to increased detoxification by metabolic enzyme, and the third mechanism can be due to target site insensitivity or alteration, and the last can be due to active excretion by ATP binding cassette transporters. So when there are four mechanisms of resistance, one is decreased penetration, second one is increased detoxification by enzymes, the third one is target site insensitivity. I will explain target site insensitivity in detail in tomorrow's class. And the last one is active excretion by APC transporters. Don't worry about this. We will be going in deep with all these things. Today we will confine our discussion only in the first two, that is the reduced cuticular penetration and increased detoxification. See, I am giving you a scenario wherein one insect is having a thinner wax layer and another insect is having a thicker wax layer. Which insect has the ability to develop resistance quickly? I will repeat. We have two insects. One insect is having a thin wax layer, another insect is having thicker wax layer. Why those who are saying thicker wax layer, they should justify why. Why it is difficult to penetrate? Abhishek, yes, it is difficult. Why? Basic reason. Anyone? Hmm? I'll take another example. I will leave insects. Yes, lipid layer acts as a barrier, lyophobic. Yes, I will take the example of colocasia leaf. Those leaves which are having very thick wax layer, and those crop which are having very thin wax layer. Those crop in നമ്മുടെ മലയാളത്തിൽ പറയുന്ന പോലെ ചേമ്പിന്റെ ഇലയിൽ വെള്ളം എടുക്കുക എന്ന് പറയുന്ന പോലെ. So those in colocasia leaves we have a thick layer of surface wax. So if you are spraying on that, the water, the spray fluid may not stick on the surface, so it will flow away quickly. So in the same manner, if the content of wax layer is with phospholipid layer is greater in the cuticle, it will contribute to insecticide resistance. So that is one way of reduced penetration, decreased penetration of the insecticide. Another thing is increasing the thickness of the cuticle. We have several examples of that, wherein the thickness of the cuticle of resistant individuals are greater when compared to susceptible individuals. What will happen when there is a change in the composition of the cuticle or change in the thickness of the cuticle? What will happen is that it prevents or slows down the toxin from reaching the target site. That sometimes what will happen is that if the thickness is more, the pesticide may not be able to reach the insect body. The target site may be acetylcholine esterase inhibitor. It may won't be able to reach the synaptic site for acting. Or sometimes what will happen is that we are told about reversible inhibition. Carbamates are having reversible inhibition. We are applying a carbamate insecticide, and the thickness is of the cuticle is slowing down the penetration of the insect cuticle of the pesticide. That means that by the time carbamate reaches the target site, it is a reversible reaction. It may not cause enough mortality of the insect. So the increased thickness of the cuticle can contribute to resistance by preventing the pesticide from reaching the target site or slowing down the pesticide from reaching the target site. Then the next major mechanism by which the insects evolve resistance is increased detoxification by metabolic enzymes. See, every insect is having a set of enzymes which are involved in detoxification of xenobiotics. Have you heard about a term called the xenobiotics? Xenobiotics. Have you heard? Yes or no? No. Xenobiotic is, I will write it fully. That is xenobiotic. Xenobiotic is anything which is not inherent to the body of the insect. That means if an insect feeds on any plant containing any secondary metabolite, say one insect is feeding on neem, say hello beltis antony. Hello tea mosquito bug is a pest of neem tree. How it is able to feed on neem tree? Because one azadirachtin is there, that is a xenobiotic. We can take it up as a xenobiotic. This azadirachtin, once it is ingested into the system, this toxicity of azadirachtin is eliminated with the help of certain enzymes present in the body of the tea mosquito bug. So tea mosquito body has its own mechanism. Every insect has its own mechanism wherein it it has a set of enzyme which will degrade or detoxify the pesticides or any compound which is coming from outside. And that is common. That's why insects are able to survive. A polyphagous pest, Spodoptera, it feeds on banana, it feeds on castor, it feeds on a variety of vegetables, and all these crops contain a wide variety of chemicals. And some chemicals, say, take the case tobacco. Spodoptera feeds on tobacco. It contains nicotine, an extremely toxic, extremely neurotoxic chemical. How it is able to feed on tobacco containing nicotine? One mechanism is it has inherent detoxification mechanisms which degrade nicotine. So we have mainly three enzyme systems which are involved in detoxification of insecticides or xenobiotic. The first one is cytochrome P450 monooxygenases. It is also called as mixed function oxidases. Next one is esterases. The third one is glutathione S transferases. So the three enzyme group are esterases, glutathione S transferases, it is abbreviated as GST, and mixed function oxidases or cytochrome P450 monooxygenases, it is abbreviated as MFOs. So these three are the major three groups of insect enzymes which are involved in detoxification of pesticides. And one thing is that I will discuss in the next class, the next example. Take the first enzyme I will take is esterases. Can you name a few insecticides which is having ester bond? Few insecticides having ester bond. Hmm? I have dealt in detail about insecticides with Estabon. Yes, cypermethrin. Yes. What about others? Resha has given one example, cypermethrin. What about others? All the pyrethroids are esters. Remember, esters of acid and alcohol. We have discussed like chrysanthemic acid, pyruthric acid, alcohol, cinnarolone, pyrethrilone, jasmine, all the then OP compounds. All are esters. Phosphoric acid. So all these pyrethroids OP are having ester bonds. So what will be is esterases are, as you know, esterases are those enzymes which breaks ester bond. And these enzymes, these are ubiquitous enzymes. That means esterases are present all over, which is involved in the degradation and metabolism of insecticide, and they degrade all those pesticides contain ester, thioester, and amide bond, mainly those containing ester bonds. It can also degrade those chemicals containing amide and thioester bonds. So, as we discussed, it is involved in the detoxification of OP, carbamates, and pyrethroids, mainly having this ester bonds. So I will discuss how insects evolve multiple resistance. Say a person was continuously using an OP insecticide, say malathion as we have discussed earlier, and the resistance development is due to increased level of esterases. That means the insect is releasing a huge quantity of esterases, and because of the increased esterase level in the insect body, all these ester bonds in the OP insecticide, that is malathion, is degraded, and the insecticide is not having any effect on the insect. Then the farmer is going for a synthetic pyrethroid. The same esterases can break the ester bond in pyrethroid. Though OP is having acetylcholinesterase inhibition mode of action, pyrethroid is acting on sodium channel. It is acting on two different sites, but the both are having similar chemical structure. Both are having ester bond. Because of the continuous use of an insecticide, OP insecticide, the ester levels in the target pest is extremely high, and the esterase enzyme, the increased level of esterase enzyme will break the ester bond in pyrethroids also. Do you understood how multiple resistance develops? Even if the two pests are having different mode of action? Yes or no? Yes. What about others? Or shall I repeat it again? Shiga, Sri Lakshmi, all are having some network problem. Rahul, repeat. Yeah, okay. Uh, Esterase, I will say OP compounds are having ester bonds. The farmer is using continuously going for sprays of an OP insecticide. And with the continuous use of OP insecticide, the insect has evolved resistance by increased production of esterase enzyme. With the increased production of esterase enzyme, this esterase enzyme will act on OP compound and it will break the OP compound. Once the OP compound is broken down or metabolized, it won't be able to exert its effect. So the farmer is applying an OP insecticide because of the high level of esterase enzyme. The OP insecticide will be broken down, metabolized, and the OP insecticide won't be effective. So the farmer is finding the OP insecticide as not having any activity to the insect. Then farmer shifted to another pesticide, that is a pyrethroid. Farmer started spraying pyrethroid. The pyrethroid is also having the same ester bond. The insect is already having high level of esterase enzyme because of the continuous use of OP insecticide. And this same esterase will act on pyrethroid, the ester bond of pyrethroids. And the pyrethroids will be easily broken down into acid and alcohol. It won't be able to exert the effect, toxicity. So, if the two metabolic pathway of two insects are similar, then the insecticide cross resistance or multiple resistance can happen pretty quickly. Is it clear now? Rahul and Sri Lakshmi. Yes, okay. Okay, then how this levels of esterase enzyme increases? What happens? I just told you that there will be increased level of esterases. You might have studied about one gene, one enzyme, etc. If there is an increase in enzyme level, enzyme is a protein. In enzyme level, that means there is a change in the gene genetic level. It can be due to gene amplification. That means the gene, the number of copies of the gene. So earlier in a genetic material of that insect, earlier if the gene number of genes was two, which you used to code for esterase, because of the development of resistance, the number of genes will increase to say 20. That means a tenfold increase in the level of esterase enzyme. So that is gene amplification can be one thing. And then there is upregulation of gene. Instead of increasing the copies of the genes, there can be a promoter sequence which upregulate. That means it make the gene to produce more quantity of the enzyme. So there can be a promoter sequence which will amplify the expression of the or increase the expression of the same gene. So that is upregulation. Or sometimes what it happen is the enzyme, there can be a change in the active site of the enzyme. That mutation in the active site of the enzyme what will make the enzyme more potent, more active. So this can be the reasons by which there will be more production of enzymes. I'll continue for 2 to 3 minutes and I will finish off. The next is glutathione transferases. Glutathione transferases is again a large group of enzymes which is involved in the detoxification of xenobiotics. So here what will happen is this is xenobiotic X. This is glutat. Just a minute. This is glutathione. This is xenobiotic or pesticide. This is reduced glutathione. You can see that SH, that hydrogen atom is attached to the thiol group. Thiol is S. So glutathione transferases. This is the enzyme. This glutathione transferases catalyze the conjugation of reduced glutathione. This is reduced glutathione with the xenobiotic C. This hydrogen is removed and this glutathione transferases catalyze the conjugation of reduced glutathione with the xenobiotic. So that means it is a conjugation reaction wherein the glutathione transferases catalyze the conjugation of reduced glutathione with the pesticide. What will happen if the conjugation happens with glutathione and the pesticide? If the conjugated product with glutathione and the pesticide will make the pesticide water soluble. And once the pesticide is water soluble, it can be easily excreted. So that is how the resistance occurs. So glutathione transferases catalyzes the conjugation reaction between reduced glutathione and the pesticide, and this conjugated product is extremely water soluble and it can be easily excreted. This is normally happening in the case of DDT, dehydrochlorination reactions of DDT. That is one major detoxification mechanism in DDT resistant insects. Coming to the last enzyme, that is mixed function oxidases or cytochrome P450 monooxygenases. Its name mixed function oxidases is because it can catalyze a diverse catalytic reactions. Diverse reactions. It is having genetic diversity. That means several genes can code for the same enzyme P450 monooxygenases. It has broad substrate specificity. That means it can act on large number of substrate. Can act on different number of pesticides. Say organochlorine, organophosphate, carbamate, synthetic pyrethroid. See the insecticide to which it has evolved the resistance to organochlorine, organophosphate, carbamate, synthetic pyrethroid, neonicotinoid, IGRs, almost all groups of insecticide resistance. One major mechanism of resistance is due to increased level of mixed function oxidases. That is because of its broad substrate specificity. That means it can act on a large number of substrates. Esterases, it can act only on those insecticides which are having ester, thioester, or amide bonds. Here it's not so. It can act on diverse substrates and it can have catalytic versatility. That means it can catalyze diverse kinds of reactions. And another thing is that it is also involved in detoxification as well as activation reaction. We have discussed about the pro-insecticide, malathion to malaoxone, parathion to paraoxone, etc. So this conversion of malathion to more toxic malaoxone or parathion to more toxic malaoxone is because of the activity of mixed function oxidases. And it catalyzes dealkylation, that is removal of alkyl group, dearylation, removal of aryl group or an aromatic ring. All those things are catalyzed by mixed function oxidases. Again, increased production of cytochrome P450 by gene duplication, gene amplification, upregulation, etc. I am stopping it for today. I think many of you have had some network problem in between. I will upload the recording in Moodle. I haven't done it for the other classes, so I will do it today itself. All the classes till now will be uploaded in Moodle. Don't worry, I forgot it in between. I will upload it so that you can view it again. If you have any doubt, you can ask it now. Anything? Rahul, Abhishek, Shika, Sri Lakshmi? No. Okay. Okay. Fine. Shika? Okay. Okay. Thank you. Thank you very much. ഇടയ്ക്ക് miss I will upload it in Moodle so you can see it. I think Srisha also had similar problems. So I will upload it in Moodle and you can watch it again and again as long as you wish. Okay.
നാളെ ക്ലാസ്സിലുണ്ടോ?
If you wish, I will take. I am free tomorrow also.
അത് വെച്ചാല് നാളെ ഞാൻ അങ്ങോട്ട് വരുന്നുണ്ട്. കർഷക ഭവനത്തിൽ നിന്ന് സാധനം എടുക്കണം. അതിന് വേണ്ടിയിട്ട് രാവിലെ അങ്ങോട്ട് പോകുന്നുണ്ട്.
ഓക്കെ ഓക്കെ ശരി 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 ആ ഓക്കെ എന്നാ വേണ്ട നാളെ വേണ്ട